a centimeter long, and then as it's been growing, or as it was growing before it was collected. Fascinating to think about.
Yep. And one of the things with all these deep sea species is there's only a few of them that we really have done any really solid growth rate studies on. So we know that some species are faster growing than others. They're kind of, you can give them in terms of like forest succession, like the weedy species that will come into areas quickly, but we really don't have solid numbers on a lot of their growth rates, and how old they can get. So that's one of the things that people are studying moving forward.
And the succession dynamics in the Pacific Islands have been interesting when they've had the chance to study lava flows, and they know how old the lava flow is. So anything that's been growing on that lava flow must be a certain bounded age, and then you can see how that community changes over time if you can monitor it, if you can afford to keep going back to that site. Interesting work.
Yeah. I know there's some groups that are interested in doing that out here using shipwrecks too, because if you know when the ship sank, and they do this in the tropics as well, it gives you a marker point for the oldest the possible fauna growing on it can be.
Absolutely. I think that's pretty relevant in Olympic Coast. It sounds like the conditions in the Olympic Coast have led to, I think, more than 200 historical shipwrecks in the area. Combination of fierce weather, isolated rocky shores, and lots of ship traffic. Lots of history there.
Jenny is pointing out on our science chat with our scientists in charge that another theme for the West Coast Deep Sea Coral Initiative is looking at change over time for these fisheries boundary changes that have gone into place. That one of the things we've done through a number of cruises, including some Nautilus work in the last couple of years, has been to survey across boundaries that are opening and closing. So we'll have reference markers for the future to go back and look and see how things have changed over time.
Absolutely, and I'm sure Liz Clark can correct me if I'm wrong, but I think there were some studies back in 2006 or so, which would have been just before or when the essential fish habitat protections were getting set up on the West Coast. And then some of those sites were revisited very recently with this West Coast initiative that the Deep Sea Coral Research Technology Program has been supporting. Very excited to see what's been happening in the time frame since. Deep sea corals and sponges being such slow growers in general, of course, you need a fairly long time frame to look at their changes, but that's 14 years, 12 to 14 years. That's a pretty good time frame.
Yep.
And that was really one of the priorities of the Pacific Fishery Management Council. They spend a lot of time trying to figure out what the best setup for protection is, what the best configuration sites and network of EFH conservation areas is to protect species like what we're looking at right now. And they want to know if that's working. What's what's going on there, and are things changing when you do protect them from trawling and with other threats like potentially longline fishing around the country. Trawling on the seafloor is what we think about the most, but longlining, especially in Alaska and many parts of the West Coast, can threaten deep sea coral habitats. Trawling especially will drag a weighted net across the whole seafloor and demolish most things in its path. So that's the thing that our program pays the most attention to. But also deep sea mining is on the horizon, and that's something that some of these habitats will need to worry about. Because of all these threats, certain areas have been protected. Most recently in January 1st, 2020, a whole lot of protected area changes, and they weren't just new closures. There were some areas that were opened recently to fishing that had been closed for a while because now the stocks have rebounded, and so now we'll see what's happening when you actually reintroduce fishing into an area that's been closed for a number of years. So all that was part of Amendment 28 that was just recently passed and has gone into effect on January 1st this year. really excited to see what kind of results we get from these studies.
It's been exciting to be part of the survey. More of these fiber optic sponges. All of these ones are growing quite low to the seafloor. We have crossed quite a diversity of habitats today, starting in the deep canyons with the deep canyon with the steep walls covered in coral and sponge. We found a methane seep early this morning. Continued up, and now we're finishing up in this muddy area, but one that's full of sea pens and crinoids. And quite a diversity of habitats. Got a deep sea sole there, missing flatfish.
Yay, we found one.
The methane seeps yesterday were, or yesterday or 2 days ago, I'm kind of losing time now, were great, but today's dive has been a great kickoff for the coral segments of this expedition. I've got down in the lower left a tiny, tiny little thorny head. 2 of them.
Wow, yeah. That is so tiny.
Oh, they are small.
It's just a little guy. They're looking at that teeny little fish down there. He's like minuscule.
Abby has chimed in on our science chat that at least for some of the glass sponges, when they're small, they tend to have growth rates of 1 to 3 centimeters per year, but they appear to slow down once they reach larger sizes.
Thank you, Abby. I didn't know that. Learning so much. This is so much fun.
Thorny head on the move there. This would be a long spine thorny head, showing off that long spine that gives it its name. You do get into a depth zone where the long spine and short spines overlap, and then you can be really hard to tell them apart unless they show you that dorsal fin. Got another little lanternfish there in the center, little black fish that's swimming around.
Excellent. Just bugged the thorny head. Reminds me of seeing birds.
Those lanternfish are in
when there's a big eagle or something like that sitting on a telephone wire and a smaller bird comes and bothers it and it actually flies away. Looks like what just happened there.
Yep, a few little lanternfish we've seen like that one. They are in the group the McCophids, and they're called lanternfish because they've got little bioluminescent spots. The spots can be different places on around their belly or around their eyes, depending on the species. They are very, very common and abundant in the deep sea, and a very important source of food to a lot of more pelagic predatory fish. There's a nice fiber optic sponge who we can't see the fiber optic cable on because it is completely covered in crinoid. I think I really like using those as a way to get up off the sediment into the water column, probably to feed.
So Meredith, what was the highlight of the day for you?
I'm gonna have to go with this big Paragorgia. I've rarely seen a stand of Paragorgia quite like that. And they were, you would just, you'd move through a little sandy patch and then just get into another round of them. It was quite impressive.
Yeah. That's a good one. I was in a training all day, so I didn't get to watch most of the day's discoveries, but I did have it on in the background. I will admit, don't tell anyone, but I was definitely watching the live feed in the background, and I definitely saw a few Paragorgia.
I had to go into town midday, so I had to leave the watch after my watch. My supervisor of the last 8 years is retiring, so we're going to meet up for a quick socially distanced outdoor lunch. It was lovely to see her, but on the ferry, I actually put the little screen up on my cell phone and had my laptop as my hotspot so I could join the science chat. What else am I going to do on a 35-minute ferry ride?
Of course. You've seen the scenery before. You haven't seen this scenery before, though.
So Jenny has proposed, as we kind of fly along here with the mud, a final introduction of the science team that's on right now, including Nav and the pilots and the data logger. So I can start. I'm Meredith Edlett. I'm with NOAA's Northwest Fisheries Science Center. I'm a deep sea coral geneticist, working with the Deep Sea Coral Research and Technology Program and the West Coast Initiative. And I do a lot of eDNA work and taxonomy.
Sorry about that.
Heather?
Hi.
Yeah.
Erin Heffron,
Sure.
navigator. Watching the last bit of our transect here.
Thank you, Erin. The navigation has been awesome. It's been great to have all of you be involved in this dive, especially since it's my first one narrating for Heather Coleman. Like Meredith said, I'm also from the Deep Sea Coral Research Technology Program. Just really happy to be here. Really excited to have this partnership that we have with Ocean Exploration Trust. Just so grateful that we can use these resources to explore the deep sea.
And next to me there is
Kim Wallace, currently ROV team, currently piloting Hercules almost as well as Jess does. Jess on my left.
Hi, I'm Jess and Duval. Sorry, forgot half my last name there. I'm in Argus currently.
And I'm Susie Cummings, and I'm currently a data logger, but I'm also working with the science team, getting ready to process some samples after this.
All right, guys, you want to take the last 5 minutes to get stretched out?
I think they have a request for a Niskin before we go.
Sure, no problem.
Science, is that correct?
Take one final Niskin right before we leave the bottom.
Yeah.
Okay. And is there a specific altitude you want that at?
Not really. So just right before we lift off. So this altitude would be great. If we need to get a little higher while you get stretched out, that would also be fine.
Sure thing. Ah, okay.
You get a lot of sea pens in this sample. One of the really fun things about doing the eDNA analysis is then I can go back and find out how many times I'm now wrong on video in perpetuity in my identification.
And you're counting that in the fun category?
Yes, it's a fun kind of wrong.
Okay, sure.
But I also review the video while I'm doing the eDNA identification, and so it is actually a very effective way to go back through and say, okay, this is what I'm seeing in the sample, and this is what I'm seeing on the screen, and kind of adjust what you expect species to look like.
Yeah, makes a lot of sense.
Susie, this is going to go in
Niskin number 6.
6, Roger.
And it will be sample number 42.
42, Roger. Would you mind looking a little bit to the left there? me get a sample?
Sorry, Susie, that was 042.
042.
Thank you.
Oh, auto depth will slam you into the ground or slam you up. So you'll want to get out of auto depth.
Doesn't work?
No.
Nice.
Sometimes if we double click it, it works, but other times it doesn't. I'm just gonna look a little left. Great, thank you. Thanks, Kim. Come on, fingers. All right, let's try that. Okay, and
Pop.
pop pipe.
Cool.
See that? It's
Excellent,
straight.
thank you.
Oh,
It's not settled in right, the lid.
no, I don't have any
Hmm.
control of that, unfortunately. That's a function of the bungee inside.
Mm.
So even if we kept pulling at it, it wouldn't actually make it release any different. Sorry, guys, that might not be a very valid sample for you.
Well, we'll make a note of it and I will process it anyway and we'll see what we get.
Sure thing.
Maybe we'll get something midwater if it snaps closed during ascent.
That's roughly hemispherical on the bottom, isn't it? I mean, it might actually seal.
Yeah.
Okay, ready for recovery?
Yes, ma'am.
Cool, I'll let the bridge know that we're ready to come up. Bridge nav. We're ready to start our ascent with the vehicles. It'll be about 45 to 50 minutes to the surface. Thank you.
What's going to be our target heading for recovery?
Right now we're into the wind at 188.
188?
Yeah, I'll check and see if it anticipates. Oh, we're into the current actually. Let me check on that.
Getting that one last lingering look at the bottom of Quinault Canyon. Taking enough samples, I'm sure that we're going to keep the science team very busy.
All right. That makes our life pretty easy, doesn't it?
Yeah, just happen to be set up just right.
I'm going to come about. Yeah, it's I think it's something with whatever PID control, some we've tried playing around with it before and it still slams. But yeah, so it's gonna jam you up now. Sometimes you can cycle it and it works. All right, I'm around 190. I'm ready to come up when you are, Kim.
Thanks to all our scientists ashore who joined us throughout the day on this dive. We'll see if any of them stick around with us for this transit up through the water column. I always actually like to see if you never know if you're going to get, how lucky you're going to get on what you see as you ascend or descend. If you get squid, sometimes you get pretty cool fish. You know, sharks a couple times.
A voluntary blue water watcher. That's fantastic.
Sorry, I just lost my connection there briefly.
Too. Yeah, that was strange.
Just as someone said, voluntary blue water something.
Right. I was just going to say how incredible it is to be in a submersible going down, down, down as it gets deeper and darker and seeing all the fascinating things go by you, and some bioluminescent when you turn off the lights, and some things much larger than you'd like them to be. Pretty fascinating.
See this blue water area they call the deep pelagic or the deep mesopelagic is actually one of the great unexplored areas of the globe. It's not on the bottom, it's not on the surface, and there's a lot of it. There's a lot to be explored in this zone.
I think I've heard 95% of the Earth's living space or something like that is a water column.
Yeah, I know people were chatting about it earlier in the dive, and we haven't seen any of the, and now I'm blanking on the species, those pelagic tunicates on this dive.
None at all. How sad. We'll see if we can get some on this transit up. You're talking about siphonophores?
No, it's the ones that bloomed like crazy 2 years ago, and they were all over the bottom and in the water column. Oh. And it's the 3 a.m. Start this morning is starting to cook my brain.
You signed up for that time.
I did. But I'm used to standing the night watches on a lot of our cruises. Much like actually the cruise in 2017 here, we run 2 vehicles, and so the ROVs are usually at night. So when I've participated in a lot of ROV watches, it's been the night shift. So I'm always willing to volunteer for the weird hour shift.
Absolutely, but then you don't have to go to work in your office during the day. You can just work at night.
Yep. And at night, my dog's asleep. So now that we're broadcasting from home, there's less frantically pushing the mute button.
Yeah, my dog's decided she wants to play, but I told her to wait a few minutes, and so she's being very patient, waiting till the end of the dive till we play.
Good to hear that.
So I wanted to take the opportunity on our trip up the water column to remind everybody that this dive has been a collaboration between Ocean Exploration Trust and NOAA. And it's been a fantastic opportunity to showcase the unique science and the education capabilities of the UV Nautilus by exploring and studying our national marine sanctuaries and the waters just surrounding them. And today we've been just surrounding the Olympic Coast National Marine Sanctuary, very close to the sanctuary boundaries. These live telepresence capabilities of the Nautilus are allowing scientists from around the world to participate and to contribute to the success of this mission. And there will be many, many, many hours of video analysis to come that will teach us quite a lot about the deep sea. We learn so many things every time we send an ROV down to the seafloor. Students from across the country and around the world are also directly engaging with Nautilus and OET staff and operations, and they're bringing the ocean and the science to their classroom. So we're really happy to be a part of this.
Yes, thank you very much to the Ocean Exploration Trust. And we're really looking forward to seeing the samples and all of the video. And I just got a very helpful text from Jenny Waddell. I was talking about pyrosomes.
Can always count on Abby. Thank you, Abby.
No, it's Jenny, but we can also always count on Abby. Well, both of them have chimed in. I was looking at two different screens. I will say that is one of the advantages of working from shore right now is the ability to still receive text messages from other scientists ashore who aren't on SPL to say, it's a pyrosome.
That is nice.
Jenny has also filled in that it is the name means pyrosome means fire body, and it is a subtropical colonial tunicate that several years ago bloomed extensively throughout the North Pacific. And you do still occasionally see them around in the summers here, but now that we're in the end of September, we're outside of the timing when we would encounter them. My favorite thing to look for on blue water ascents is intact larvation houses. Larvations they're in the chordata. They're kind of distant related to tunicates and other species, but they're pelagic. And they form these houses that they build out of mucus that actually have very intricate, delicate structure to them that they use to filter feed. And they will suck water through them for a day or two until they become too clogged, and then they discard it and build another. But they're quite striking when you get a full intact one on camera.
Is it something you've only seen like once or twice, or do you see them fairly frequently?
You see them fairly frequently. And you'll actually see them kind of collapsed and full of marine snow. They're actually a major source of carbon transport to the deep ocean, and these days a source of microplastic transport to the deep ocean. There's one right there. That's a collapsed larvation house. It's just going past the lasers. But they're just really pretty to see when they're completely intact. There's some fun microfluidics research going on right now using lasers to kind of track how they move water in an efficient manner for filter feeding.
Must be a really tasty treat for whoever gets to gobble one up when it's full of stuff that it's accumulated.
Mm-hmm. Okay. That lands right near you and you're a coral. Got a lot of food.
Yay! If you can even eat something that big.
Yeah. Well, I mean, they're made out of mucus, so.
Compressible.
Oh, there's a pyrosome. They are here. So the pyrosome was not the kind of frilly white that just went through. It was the kind of purple oblong object.
Your video is just half a second faster than mine, so it's like you're seeing the future. Kind of fun. Chalking it up to you being so much closer to the actual site of the ship and maybe being all the way across the country, but I doubt that matters.
It probably makes a minor difference. I've also found through the day today that as the day has gone on, sometimes my video gets like half a second slower, and sometimes if I pause and restart, it catches up again.
That makes sense. Was that a jelly that went by about a minute ago?
That was actually on some dives in these deep canyons. Occasionally, you will get into these big swarms of the jellies, and I've seen very few today.
I've seen enough recently. The worst thing the ocean has ever done to me was about a month ago. I was in South Carolina and I got a massive jelly sting on both my calves. It was really bad with tentacle marks all over. A month ago, and I still have the marks, although it doesn't itch anymore. But I don't know what I did to the ocean to deserve that. Maybe all my experiments in undergrad torturing sea urchins. I'm not sure.
I've only been stung once, and that was scuba diving here in the Pacific Northwest. And the thing about scuba diving here is you're so covered in neoprene and dry suits. The only place you can be stung is on the face. But then the water is so cold that it only matters for about 10 seconds.
Yeah, I've been there. I actually got one stuck in my wetsuit once. That was pretty miserable. At least it was not a super stinger like this one. It's worth it. The ocean is such a fantastic place full of so much to discover. An occasional jelly sting. I'll take it.
Yep. I think we've given the Southwest Fisheries Science Center team plenty of transects to analyze through the day, through the next little period once they get the video.
You're welcome, Science Center.
And I'm really excited to see what some of these sponges and corals we've collected through the day have turned out to be. I think the coral collection that I'm most excited to get a look at is the bamboo coral from early this morning. We don't get many of the single stalked variety. And that one looks like it may actually really be Lepidopsis, which would be great because that is one of the species that we are currently missing from our DNA barcode collection.
Excellent. How long does it take to create a barcode for a particular species?
It depends on the species and how much it likes our DNA primers. So when we're creating a barcode, we have these little fragments of DNA that match segments of DNA from the animal. And we use a technique called PCR to amplify about a 500 base pair fragment of DNA from the mitochondria. And it's pretty unique to species. It's not perfect, but it does allow us to identify most of the species. And so start to finish, to get a new barcode, if the primers work perfectly on the first go, is about 4 days from DNA extraction to sequencing, 2 days if I'm in the lab just full time. But it's usually about 4 days. But with a lot of these species, there is a little variation in where our primer site sits. And so we end up having to do multiple rounds of PCR to just adjust our conditions, our temperature conditions, to get that DNA fragment to really sit down and bind well. And then in octocorals, which is what the bamboo corals are, there are now 6 different known mitochondrial genome arrangements. And so that's the order of the genes in the mitochondria. And so the primers we use have to vary depending on family. And for most bamboo corals, we know what we're looking for, but we do get surprises occasionally where we can't amplify our fragment, and we can't amplify our fragment, and we can't amplify our fragment. And it's because the gene order is different, and we have to use different primers.
Yeah, that makes sense. Do you ever get the perfect primer on the first try and it really takes you 2 days?
Once in a while. Some of the Plexarids actually are very good primer matches. The time-consuming part is the DNA extraction itself, and then the PCR reaction takes about 3 hours, and then we sequence it overnight. So if I just, get in really early in the morning, finish the DNA extraction, get a PCR right away, start the sequencing, get it on the sequencer the next day, it can be about 2 days, but it's rarely that start to finish. And it's also just, I'm also answering emails, and I have other projects that I'm analyzing data for.
Yes. Very efficient.
And for the DNA extractions, it usually takes me a little while to cut the tissue. And when I'm working with the samples, I usually like to really take a close look at them. I'll often look at them under the microscope before I extract DNA, just to make sure that what I think they might be is close to what they might be. So I do take time to do some pre-sorting, looking at them on the microscope and that kind of thing as I'm working on them in the lab.
That makes sense.
Nice big siphonophore just floating by.
Yeah.
Nice to watch. We've got a graph we have accessible here for all the scientists at home, where we can see the Hercules depth, the oxygen saturation, and the water temperature. And the water temperature is this nice, steadily climbing slope that is climbing at the same rate that the ROV is coming up through the water. We've been down at 2 degrees just above freezing for most of this dive, and we're now up to a mighty 5 degrees. That's centigrade. So one of the things that the crew cross, it's very cold. One of the things that the crew crossing the samples has to look forward to is part of removing the samples from the bioboxes, where because the bioboxes are sealed, they've basically trapped that 2-degree water, and it's not going to have a chance to heat up. And so when you go to remove a sample from these big bioboxes, like, put some gloves on, because it's going to be very, very cold.
Yikes. I don't know if I've felt 2-degree water before. That's pretty crazy.
Very, very cold.
Mm-hmm.
So once they're processing the samples on board, we have a variety of different things that are going to happen. The corals and sponges are going to be carefully photographed. Any associate species, so like little brittle stars and things that are growing in the sponges, will also be sampled, although they'll be cataloged separately from the corals themselves and sponges. Some subsamples will be taken to go to different research groups who've been coordinating with Nautilus on getting some samples. And then the corals and sponges will be either frozen or preserved in alcohol, and that allows us to work with them in the lab later. And then the sediment cores are going to be, I think Andrew's sponge for the evening. They usually section those kind of a centimeter at a time and through some fine screens to sort out all the different critters that are living in them, detailing things like the density of the sediment. So sediment cores take quite a while to process.
What do you think takes more time to process, a sediment core or going through the video of the benthic video?
I think overall the benthic video, but that's just because when they're really doing the analysis, it takes the same amount of time as the dive, plus a little if they rewatch 5-minute segments. But in terms of shipboard sample processing, sediment cores are definitely one of the longest.
Yes. Participated in a few undergrad years.
So actually for the sediment cores, for the more experienced scientists who are, you know, have a hand at doing the sediment coring, they can be pretty quick, especially if you've got multiple people working on it at once. Like Andrew was showing me and Leila how to do the sediment core stuff, and Andrew's pretty fast himself because he's done quite his share of sediment cores over his career.
That's good. I'm not fast at sediment cores.
It really depends on I guess what method you're using for it. Like Andrew has this mechanism that he made out of PVC pipe basically, where it extrudes the core and then you can just take a slice off the top and then just chuck it into a bag or into preservative media, depending on the sample type.
Oh, nice. So you're not having to put it through screens in situ on the ship.
Not usually, although we will do some animal processing stuff where we're going to be sorting it via microscope and that sort of thing. But most of the stuff that we're focusing on for this cruise is either chemical analysis or microbial analysis. So those things are just like basically put it into the freezer and then we'll do stuff with it later.
Save it now, process down the line. Yeah, I'm excited for some of our subsamples. We've been talking with Andrew and some of their lab group, and they're interested in running the metabolomics on some of the corals and sponges. And so we're pretty excited about that. So we'll be doing some of the genomic stuff, and some of the species we're going to do actually rad sequencing. So we'll have some whole genome capture, and it'll be interesting to compare the outputs from those two processes.
Yeah, I'm actually pretty excited on that front too, because I'm doing the same thing with the microbial samples. I do a lot of genome sequencing, and now we should be getting some RNA samples from our stuff too, so we can do some transcriptome processing. And then Carrie McPhail's group will be looking at the metabolomics of the sediment samples that we have as well. So we'll have all of these with the same sediment slices, basically the same depth into sediment sections. And then we can compare that information.
You want to explain really quickly to everyone who's watching online what metabolomics is? Because I'm sure plenty of people have not heard of it.
Yeah. So metabolomics is, some folks might be familiar with kind of the idea of omics, but if you're not, it's basically about taking large amounts of information from the environment about kind of the genetic coding of things. So metabolomics is about targeting kind of the the chemicals that are produced by different organisms within certain environments. And so that can be done from microbial sources, which is what I focus on, and Carrie McPhail's group focuses on, and many other groups focus on. But it can also be done from animals and other creatures like sponges.
Thanks, that was great.
Thanks.
It'll be interesting to see also with samples that are being collected coastwide, we'll collect multiple samples of the same species, whether the metabolomes change as you get into different areas of the coast.
Oh yeah, that's super interesting to us as well. I think since we focus on methane seeps in our research, one of the ways that we're doing those sorts of comparisons is by looking at samples that are right at the methane seep and then samples that are physically further out from the seep at different distances. And so sometimes we can see different sorts of patterns as things are closer or further away from the methane seeps.
That's really cool.
Yeah, I'm excited.
Mm-hmm. Some of the sponges we've collected through the West Coast Initiative, we're interested in seeing actually also what chemical compounds they may be producing for biopharmaceutical research. So we've been looking into that, hoping to find some partners to work with. We've got sponges in the freezer.
Yeah. Yeah, that's super exciting work. Actually, as part of my master's degree, I worked in the lab of Dr. Bill and Lena Gerwick at Scripps Institution of Oceanography at UC San Diego. And they're doing that same sort of exploratory research for biopharmaceuticals within cyanobacteria, basically blue-green algae, at different coral reefs around the world. And so that's how I sort of got into this topic. And so it's been an area of interest for me ever since then.
I actually worked in the world of RNA as a graduate student at the University of Miami, looking at how coastal fish populations respond to low oxygen, doing microarrays instead of RNA-seq.
Yeah, very cool.
How do coastal fish respond to low oxygen?
Well, it depends on the fish. The species that we were working with was a the Gulf killifish, Fundulus grandis. They're called mud minnows. They live in estuary environments on the Gulf Coast, and they are very tolerant to everything. Those are fish that will survive. They're kind of like the cockroaches of the sea. So when we did our low oxygen challenges, within 24 hours, the fish would be behaving completely normally, feeding completely normally, and actually occasionally spawning in the tanks of low oxygen. So they do quite well, and they do it by completely rearranging their metabolism. Other species are far, far less tolerant. In an area where you get hypoxic zones, you can get species like that are quite, they take advantage of the low oxygen, use it as kind of a niche separator, but you get a lot of other species who will basically either flee or die.
Are fish, or at least the killifish, kind of like sea urchins, and that when they get really stressed out and it seems like maybe it's the end for them, they spawn?
In these ones, no. This is just, we were doing the experiments in the spring, and they were like, it's spring. So they spawned in our tank.
It was time. Okay.
Yeah, it was time.
Meredith, should we remind people?
Oh, go ahead.
I'm just going to ask if we should remind people about Olympic Coast Sanctuary and where we're going to be exploring after the weather dies back down and the ship can get back out to ROV operations.
Absolutely, that's a great idea.
Okay. Well, the meteorite, the first dive, that was actually in sanctuary waters, right?
Yes.
Okay, so we started out in sanctuary waters, and today we were just outside the waters. But the Olympic Coast National Marine Sanctuary was established in 1994, and I would love to be able to ask these as trivia questions to people and see if they could guess the right date. But I don't really have that capability, so I'm just gonna have to say the facts. It encompasses more than 3,000 square miles of coastline and coastal ocean off Washington State. There's quite a bit of area and quite a lot of seafloor underneath that. The sanctuary extends in some places to 50 miles seaward and covers a lot of the continental shelf and major submarine canyons. It's, as we've been talking about, protects a productive upwelling zone. We spoke a lot about the old water of the Pacific Northwest and how that's starting to come back up to the surface with all the nutrients, and that really nutrient-rich water is home to a number of marine mammals, seabirds, all kinds of other creatures. So on the west coast, where we are now, the seafloor gets deep so much more quickly than the East Coast. So I'm sitting in Washington, D.C. right now, pretty close to it, where we have lots and lots and lots of shallow water, and then we have these submarine canyons that dive deep down quickly, but it's quite different in the Olympic Coast and areas nearby, where much of the continental shelf and heads of the 3 major submarine canyons in the sanctuary, they do reach depths of 1,400 meters, but they get there much more quickly than on the East Coast, which I find kind of fascinating, the difference between them. See, they also have some of the most diverse kelp beds in the world, which is impressive. We're going back on the shallow side. I've done a lot of research in kelp beds and find them fascinating, so it's always fun to consider. The rocky shores of the Olympic Coast have some of the highest biodiversity of marine invertebrates and macroalgae of all Eastern Pacific coastal sites, and that's going from Central America to Alaska. It's a huge range, and it's really impressive. So we have a lot to protect and a lot to be thankful for in the Olympic Coast National Marine Sanctuary. And we've only so far in the first ROV dive of this mission explored a tiny little bit of it, but hopefully this rough weather will not last too long and we can get back out to see more of its seafloors.
Yep, and we should also mention that the Olympic Coast National Marine Sanctuary is also houses some of the usual use areas for Washington State's 4 coastal treaty tribes. And we'll be doing a couple of our feature dives within some of those usual use zones, and it's been a great opportunity to partnership with the tribes for some of this research as well.
Absolutely, and the tribes, many of them were involved in the process we set up at the beginning of the West Coast Deep Sea Coral Research Initiative. And back in 2018, they were involved in our priority setting workshop, where we got lots and lots of interests together, particularly the Pacific Fishery Management Council, but also different parts of NOAA, other government agencies, universities, NGOs, industry, whomever is interested in deep sea exploration, deep sea research, what's on the seafloor, any of these things, to share priorities and see what we had in common, what interests we have in common, and what kind of research we could do together collaboratively. As we've mentioned, deep sea research is incredibly expensive, so if we can work together, it's much easier, makes the dollars stretch that much further, and we learn that much more because we're able to form these partnerships. So the tribes many of the tribes were involved in that process, and that is just wrapping up pretty soon. Starting on October 1st, we'll have a ramp down year for the West Coast Initiative, where we'll be asking our researchers to do a lot of data analysis, all this data that we've collected. We have to learn what did we see. There's no point in just taking all these hours of video and then letting them sit on a shelf. We want categorize it and learn what kind of new species are here. And Meredith's identification and also the water samples containing eDNA are key to that process of figuring out who's living in these sanctuaries and other waters along the West Coast. So we have lots more work to do, but the research part of the initiative is wrapping up pretty soon.
Yep, it'll be a big data analysis year, cranking through all of the samples we've been able to take and the video transects. I'm really looking forward to those last 2 years, we've started trying to really pair the environmental DNA samples to the transects to see if we can incorporate that as a new tool for the quantitative transect process. So we're going to decide to see how that goes. We're now up to 313 meters, so only 250 meters to go. So we'll be at a kind of 50-meter threshold point where the ROV team will really get settled in and get set up for recovery. Recovery is a complicated process, so we've already got crew out on deck and getting ready to get set up for recovery now, going through the plans and getting people and equipment where they need to be. I've been watching as we've been chatting through the water column, and I have yet to see an intact larvation house, but I still have hope. There's still time.
I knew you'd be watching. I just learned that there are 29 species of marine mammals residing in or migrating through the Olympic Coast National Marine Sanctuary. That's impressive. That's a lot of different kinds of mammals. Have you ever, Meredith, have you ever seen a marine mammal on the ROV screen as you're sending or descending?
I have seen sea lions about twice.
That's fun. Were they playing with the ROV at all, or just doing?
They're just kind of doing flybys.
Yeah, as they do.
My favorite thing from previous years is a lot of times if they're doing dives during the day, albatross will kind of follow the ROVs around, and they'll land basically on the ROV on the surface. And if you watch the cameras as they're doing recovery, you'll often see all the little feet from right under the water surface, especially if it's very calm.
Feet. That's the best. That's fabulous. Can you distinguish an albatross foot from another type of bird foot?
No, I am not that level of bird watcher.
One day.
I only know they're albatross because I can look at the outside cam and say, okay, brown albatross.
Actually, if you're on the ship, it's not too hard to distinguish albatross from other things because they have much, they're bigger and they have really long, thin, streamlined wings.
But can you ID them by their feet?
Sounds like no. We'll have to work on that, Meredith.
Jenny is commenting that, Ocean Exploration Trust several years ago when they were doing work in the Gulf of Mexico has a great video that's available online where they had a young sperm whale come in to visit the ROV in midwater, and it just hung out for a bit. So if you'd really like to see some cool video footage of a sperm whale, look that up from the Ocean Exploration Trust. It's quite amazing.
Oh, fun.